Hey, good morning. Today I want to continue our discussion of GMO foods with some important and historically relevant examples. Okay, so just as a reminder, when you're going to modify a plant, you need to use plant transformation, and there are two major ways in which that is done. One is using the bacterium, agrobacterium, its tDNA, to transfer the gene directly to the plant cell, or you can use particle gun ballistic method, in which essentially a gun has little teeny particles, metal particles, that are coated with DNA that are shot into the plant. And the cells that survive are likely to have taken up your molecule. Okay, after that, the procedure is really pretty similar. You need to select for the cells which have acquired the plasmid or molecule of interest, grow them in culture, and then regenerate the plant from those cultured cells. And so this can be a long, drawn out, and fairly difficult process, but that is still in use. Okay, so in the case of the agrobacterium, just to remind you, the part of the plasmid, of the TI plasmid, that is of relevance is what's called the tDNA, and it's flanked by two sequences, one called the left border and the other being the right border. And the DNA in between those sequences was actually transferred to the plant. The rest of the TI plasmid remains behind. And these, of course, have been carefully engineered to remove all of your cell proliferation causing genes, etc. But they do have essential things like selectable markers for E. coli and for agrobacterium, which is needed, of course, for the cloning purpose. Okay, so our first example is going to be the glyphosate roundup story and traditional transgenesis. Okay, glyphosate is an herbicide. Now, it's been in use since the 1970s, long before we had the transgenic plant versions. Okay, and it's a good herbicide. It's very soluble in water, and it targets an enzyme that plants require for growth, which animals do not need or have. Okay, the enzyme is called EPSPS, or sometimes EPSP synthase, or 5-enol pyruvyl shikimate 3-phosphate synthase. So you can see the need for the abbreviation. Anyway, in plants, this enzyme is required for the synthesis of aromatic amino acids, phenylalanine, tyrosine, tryptophan, which are also metabolized further to produce molecules that are required for photosynthesis by these plants. So obviously, the plant needs this enzyme in order to live, and people don't because we get these amino acids from our diet. Okay, so here's our pathway, which is blocked by this glyphosate intermediate. And as a result of that block in the pathway, the aromatic amino acids are not made, and the plant cannot survive. Okay. So many crop plants now have been engineered to be resistant to glyphosate. The first was soybean, but others soon followed, corn, cotton, sugar beets, etc. And what they have been engineered to contain is a bacterial form of the enzyme that is not inhibited by glyphosate. Okay, the enzyme is called C4 and was cloned from an agrobacterium. Okay, so this is not a manipulated molecule. This is a naturally occurring molecule, which has been then introduced into plants to allow the plants to survive the presence of the herbicide. Okay, so here's what the original cloning vector looked like. It has all the things you would expect it to have. It's got a selectable marker. It's got an origin that works in E. coli. Importantly, it has actually two copies of the CP4 gene, both of which are driven by strong plant promoters. And as well, they've been engineered to contain a transit peptide, which actually originates from petunia, in order to get the protein into the chloroplast, where this pathway actually exists. Okay, so it has all the essential things, including promoters, terminators, whatever, that are needed. Okay, so this basically summarized, I got this from somewhere, basically what's in that particular molecule. All the things you need, plus a few other things we have not actually discussed. And you do need selection for plants, which is the MPT2 neomycin resistance gene. Okay, so Roundup is a herbicide which contains it's not really what is an herbicide, but it contains a glyphosate, but it also contains some other compounds, which turn out to be really of concern. Okay, here's our pathway again. Okay, so the glyphosate's the active ingredient, and it has been wildly successful. These Roundup-ready plants can survive in fields that have been treated, and the weeds, of course, all die. So plants that are resistant to a variety of herbicides, not just glyphosate, are now available. But the days of wildly successful are numbered for a variety of reasons, largely having to do with the appearance of herbicide-resistant weeds, unfortunately, and also the issue of whether the Roundup is actually an active carcinogen and more toxic than was originally realized. Okay, so the problem is the formulations, that the glyphosate itself is not toxic to mammals, but Roundup is not made just from glyphosate. It has other ingredients that are actually more toxic. Okay, so the idea here is that the water-soluble molecule glyphosate is not taken up well by the plant cells, right? What you need to do is get a system where you can get it past the lipids that are going to be involved in the membranes to get it into the cells. So that's why we have these other molecules, which basically serve as surfactants in order to get the actual active ingredient into the plant. Okay, now these are kind of nasty looking molecules, actually. And what we're showing here is the original one, which is used much less now for reasons of obvious toxicity. But these are, in fact, toxic to aquatic organisms. So it's not as safe as it was originally believed to be. Okay, so newer molecules, newer formulations include less toxic surfactants, but nonetheless, they're still present. And so when you read about Roundup resistance or Roundup causing cancer, it's really hard to know what component of the herbicide formulation is actually causing the problems. Okay, so is Roundup a carcinogen, or is glyphosate a carcinogen, or both together? Okay, there have been some fairly convincing studies that link high exposure of Roundup to cases of non-Hodgkin's lymphoma, specifically among agricultural workers who are exposed to these kinds of things a lot. So in some cases, these are attributed to exposure to the glyphosate. Now that may be true, but it may not be true. And I myself am not convinced one way or the other. Okay, but Monsanto did settle an $11 billion case giving damages to cancer patients who have had high exposures to Roundup. Okay, so the other emerging problems include the resistant weeds, possible environmental impacts, and the fact that maybe new approaches to weed management are needed now. The glory days of Roundup may be over, and people are working hard on getting better weed management strategies at this point. Okay, moving on to our fruits and vegetables story. One particular story I like is the fact that a lot of our important vegetable and fruit crops are sensitive to viruses of various sorts. Okay, and one case that's gone on for quite a long time, starting off in the 90s, is resistance to plant viruses, which really can devastate certain crops. Okay, so the earliest experiments in the 80s involved tobacco mosaic virus, which infects tobacco, obviously. And this is a model plant because it's very easy to work with. And a lot of the really historical work has been done using tobacco and tobacco mosaic virus. Okay, so the motivation for this work really was that people had noticed a phenomenon called cross protection. Okay, in cross protection, a weakened form or wimpy form of the virus was used to infect plants, and somehow its pre-exposure of the plants to this limited virus protected it in some manner from later infection by a more virulent form of the virus. So this was called cross protection because it protected the plant from infection. And it was thought it was the coat protein of the virus that interfered with the replication of the virus. So the idea was we're going to make transgenic tobacco that express the coat protein, and that's going to prevent subsequent infection by the virus. Now, jumping ahead a few years, it turns out that it was probably the RNA produced from the coat protein gene and it triggered an RNAi pathway, not the coat protein itself. Okay, so this is a picture of a plasmid from the really classic experiment involving the first transgenic tobacco, which demonstrated that resistance to the active tobacco mosaic virus could be achieved by expressing the TMV protein, coat protein in the plant. Okay, and then this is a standard vector. It's got your cDNA encoding the coat protein. It has a promoter driving it, plus the other stuff, left and right borders, etc. All right, so this plasmid expresses the cDNA that encodes the coat protein from TMV under the control of a 35S promoter, the cauliflower mosaic promoter, which we've seen before many times. It was transformed into agrobacterium, and they used leaf disc cuttings traditionally to transfer the tDNA from this plasmid to the tobacco cells, selecting for catamycin resistance. Okay, then they regenerated the plants, checked for the presence of the transgene and expression of the coat protein gene, and then they self-fertilized them to get homozygous lines, and that one that's expressed good amounts of the coat protein. Okay, so here are some of the results from these early experiments. What you're seeing is plants that have been exposed to different amounts of the virus. These are either transgenic or non-transgenic. The darkened bars are the non-transgenic plants. The clear bars are the transgenic plants. And you're using 3 different amounts or levels of the virus infection and carrying out your experiment for 19 days to look for signs of infection. So at the highest level of the virus, which is the active TMV virus, the non-transgenic plants started showing problems even within the first few days, whereas the transgenic plants showed very little problem. Now, over time, the transgenic plants did show symptoms, so the presence of the transgene was not completely protective. On the other hand, it very much delayed the onset of symptoms in these plants. Okay, more encouragingly from this, when you had lesser amounts of the virus inoculum, again, the non-transgenic plants started showing symptoms almost immediately, but the transgenic plants were very much delayed in when they showed symptoms, if they showed symptoms at all. Right at the end of this study, some of the transgenic plants had started showing symptoms, but many of them had not at all. So clearly, this presence of this coat protein, expression of this protein gene, does have a protective effect on the plants from this particular virus. So I think this is what I just said. At the highest concentrations, there were significant symptoms, but clearly there is a protective effect because at the lowest concentrations, most of the transgenic plants never showed symptoms within the 19-day exposure. Okay, so as a result of this, coat protein transgenes became very popular for developing virus resistance in crop plants to various viruses, including zucchini yellow mosaic virus, cucumber mosaic virus, watermelon mosaic virus, and papaya ring spot virus. So this coat protein thing seemed to be the path to take to try to create these virus resistance transgenic plants. And it was very successful. Okay, so meanwhile, it turns out, or has turned out, that the cross protection that was observed did not really have to do with the protein itself, but to the RNA. So it's now believed that most cases of this strange protective phenomena occurs because the RNA is eliciting an RNAi response. So it's the RNAi that's shutting down expression of the viral proteins and inhibiting infection. So all of a sudden, RNAi became the thing to do to try to create protective plants to protect them from viruses. So one was in papaya. Now this papaya ring spot virus is a poster child for devastated crops. It nearly devastated the entire papaya crop in Hawaii in the early 90s and has been a persistent pest throughout the world. Okay, so the original first successful experiments did use a coat protein, but later RNAi was developed in China that used a conserved region of the coat protein to trigger the RNAi. Okay, so this was introduced by agrobacterium tDNA transfer again. Cells in the callus were selected, and then plants were regenerated. Then PCR analysis was used to show the presence of the transformed molecules in the new plant. So here's what our molecule looks like. We have the left and right borders at the end of the tDNA. We have a strong promoter to drive expression, not of the protein, but of a sense and antisense version of the RNA. So that sense and antisense versions of part of the co-protein RNA are expected to form a double-stranded RNA structure in the plant that would then generate an RNAi response to similar molecules. Okay, of course, we also have a selectable marker with a promoter. Okay, so this shows some of their PCR results, where we're looking at, first of all, just the molecule by itself, the molecular weight standard, rather, the plasmid by itself, and then a series of transgenic and non-transgenic lines. Okay, so what we're looking for is a PCR product that arises from the coat protein gene. So there's two different versions of the PCR product, and so a band will indicate that that protein coding gene is in there. Right, the negative controls are negative, and so this looks encouraging that the coding gene is actually present. Okay, so here are some of the really interesting and good results that they found. These are qPCR experiments using infected transgenic and non-transgenic papaya leaves. And what we're looking at is to see whether the infection is basically halted by the fact that you're making this RNAi version of the papaya ring spot virus. Okay, so it's a little bit of a complicated slide, but not too bad. They're looking at 3 different versions of this particular virus, 1, 2, and 3, and a mix of the two. Okay, the control on the left is just the inoculum itself, which is not really part of the experiment. Okay, but importantly, along the x-axis, we have groups of samples which either are non-transgenic or transgenic at different days after infection, and they're showing the individual viruses as well as the mix. Okay, so the first group is a non-transgenic at the before inoculum, or very first, just at the beginning of inoculum. So of course you have barely detectable, no detectable really, amounts of the RNA that you're looking for, which is the AC protein. But then after several days, we are getting increased infection, where that RNA is starting to shoot up in the non-transgenic plants. And there's a break in the curve because we can't get them all on the same scale. Notice you're going up from 20 to 30,000 or something. So, of course, during infection, there's a gigantic increase in the production of that RNA, which indicates an active infection. Okay, but encouragingly, the later samples with the transgene show no increase whatever. They remain at basal or barely detectable levels in the presence of the transgene. So clearly, the presence of this RNAi is protective of these plants from the virus. And this sort of approach, along with the coat protein approach, really has totally saved the papaya trade. Okay, so here's an example of RNAi that was used to protect cassava, which is an important plant in the tropics. And you can see the necrotic lesions in the plants that are not transgenic, whereas the transgenic plants give perfectly healthy food. So this is a very important approach. Okay, so now we've moved on to CRISPR and genome editing, and some of these approaches are really, really encouraging, in that CRISPR has really taken over the plant transgenic field because it's very efficient, easy to use, etc. And there are newer applications which are very specific, such as base editing. Moreover, some of the new technologies, which I love, are DNA-free, meaning that there's no new nucleic acid permanently added to the genome. So these are not transgenic. They have been manipulated by DNA methods, but they're not actually transgenic. So it is hoped that these new technologies will lead to broader acceptance of genetically modified foods, which has been, of course, a problem all along.